Assalamu alaikum, Bismillahir Rahmanir Rahim. I'll be talking about a project that combines biology and physics, so specifically super resolution imaging and theoretical physics, in order to understand how embryos elongate. So the basic problem that we are interested in understanding is how embryos, they go from a stage where initially they are a ball of cells, and then they elongate to form this head-to-tail axis in the embryos. So we are interested in understanding what are the cellular, how cellular movement as well as the molecules, how they are organized within the cells contribute to this process of elongation of the embryo. So why study embryo development? It's a pursuit, it's an intellectual pursuit that has a very deep intellectual tradition and history. In the Muslim world, there are many verses. The scientific inquiry has been motivated by many verses in the Holy Quran. So I present this one verse, chapter 23, verse 15, where Allah Ta'ala talks about during embryo development, we go from a clot and then to a lump, and then how Allah Ta'ala says that He is the best of creators. So this is a field of study where we can reflect on the attribute of Allah Ta'ala as the best of creators. And biomedically, this is an important issue to study, because if you look at these show the death rate in per 100,000 people across these 3 years. So if you look at the birth defects, the death in newborns due to birth defects, shown by this line, it's greater than Alzheimer's, stroke, diabetes, and flu all put together in adults. So there are a lot of defects that come about in newborns due to improper elongation of the embryo. And how do physicists fit into this? The approach that we take is motivated by this saying of Aristotle. He said, to be ignorant of motion is to be ignorant of nature. So I will talk to you about how we can look at the movement properties of cells in order to understand the embryo elongation. So particularly the type of cell movement that I study is called convergent extension, where cells that are initially distributed laterally like this, so all these blobs that you see here, these are cartoons of cells, then they come together in order to elongate in this direction. So here is a video time lapse of an embryo. Initially it goes from a spherical shape in order to elongate, and then the structure that initially later forms the neural tube of the embryo. So using super resolution, these are from my collaborators in the molecular biosciences department. They can use super resolution microscopes which have very high resolution of up to 65 nanometers. So this is a snapshot of the cell zooming into individual cells. There's cell 1, 2, 3, 4 shown as a cartoon on the right here. So what happens in this type of movement is that these gray cells, they come together, they meet, and then so that they elongate in this direction. So if in this video, there are two molecules, the proteins that are marked. One is cadherin, which is shown in blue, and another is actin, which is shown in orange. So I'll be focusing on cadherin, which is a cell-cell adhesion molecules that keeps the cells together. So you can see in this time-lapse video where these two cells that are initially far apart, they come together. So what we did was, so these are the proteins that I'll be focusing on called cadherin, the cell-cell adhesion proteins. They can, this is from one cell, this is from another cell. They merge together and they bind together in order to mediate, in order to keep the cells together. So we studied how these proteins cluster across the cells. So this is showing a cartoon of how the clustering of this protein cadherin occurs. The mechanism of clustering has been studied, but its functional consequences are unknown, particularly so in embryo development. So based on our predictions that the clustering of cadherin is important, our collaborators, they did experiments. So these are embryos at a particular stage in development. These are control embryos. When they knock down cadherin, it fails to elongate in a proper way. And then you can rescue it if you have cadherin that is knocked down, and then the embryos will elongate. But then if you mutate so that the cadherin does not cluster, the elongation is again disrupted. So that's quantified here, showing the length to width ratio, the length to width of the embryos. And you can see that if the clustering is mutated, there is disrupted elongation of the embryo. And here we want to show that even though the clustering is prevented, but it still maintains the adhesion between cells. So the clustering itself is very important for the embryo to elongate. So just an overview of future research plans. We develop simulations to look at, this is another research project where we look at, this is a cross-section through a tumor that is developing. So we are interested in studying how cells move when the tumor grows and it invades into the surrounding tissue. So as a topic of research, there are new types of imaging technologies that are developing. This is light sheet, a particular imaging technique called light sheet imaging of a glioblastoma embryo, tumor spheroid. You can see the nuclei that are marked and the cells that are moving. So we are interested in understanding how do cellular level forces contribute to the movement of cells. So inshallah, I'll be starting my lab at Augusta University to further pursue this work. And this is the acknowledgement to my experimental collaborators from Wallingford Lab, and then my postdoctoral mentor, Dave Turmalai.
Jazakallah, Kakkada.